Welcome to the Biochemical Virtual Laboratory for Microbiology. Today we are going to perform several biochemical tests: the Catalase Test, Coagulase Test, Oxydase Test, Rapid Strep A Test, Latex Agglutination Test, and Check 2 tests. We're going to start with the Catalase Test. The Catalase Test is used to differentiate Staphylococci, Catalase Positive, from Streptococci, Catalase Negative. The enzyme catalase is produced by bacteria that respire using oxygen and protects them from the toxic byproducts of oxygen metabolism. Catalase positive bacteria include strict aerobes as well as facultative anaerobes, although they all have the ability to respire using oxygen as a terminal electron acceptor. Catalase negative bacteria may be anaerobes or they may be facultative anaerobes that only ferment and do not respire using oxygen as a terminal electron acceptor, such as Streptococci. Let's start our experiment. We're going to first click on the hydrogen peroxide bottle, or H2O2, and drag it to the glass slides. We're going to place 1 drop of hydrogen peroxide on each of the slides. Now we're going to use a stick and pick a colony of bacteria. Be sure to pick a colony that's off by itself. Now that we have a colony of Streptococcus pyogenes, we can add it to our slide. We see that the test is negative because there is no bubble formation. So Streptococcus pyogenes must be negative. Our stick is now contaminated, so we must throw it away in the biohazardous waste and choose a new stick. Let's choose a colony of Staphylococcus aureus for our next slide. Here we see bubbles are produced. This means that Staphylococcus aureus is catalase positive. Remember to dispose of your stick properly. Contaminated glass slides should go in the biohazardous sharps container. Great, we can move on to our next test. Our next test is the Coagulase Test. Coagulase is an enzyme produced by Staphylococcus aureus that converts soluble fibrinogen in plasma to insoluble fibrin. Other Staphylococci do not produce coagulase. Thus, this test can be used to distinguish Staphylococcus aureus from other strains of Staphylococci. Let's begin our experiment. We will first click on the water bottle and drag it to the slide and place 1 drop of water onto the slide. We'll go ahead and place 1 drop of water on the other slide as well, to be used later. Now we can pick our colonies. Let's pick a colony of Staphylococcus epidermis. We'll use a colony that's nice and round off by itself, and let's add the bacteria to our slide. Our stick is now contaminated and must be disposed of in the biohazardous waste container. Now we're going to add rabbit plasma. Now we're going to gently mix our slide to see the results. We see no precipitation, so this is negative. Let's dispose of our slide in the biohazardous sharps container. Now, let's add Staphylococcus aureus to our second slide. And dispose of our stick. We'll add our rabbit plasma and gently shake for 15 seconds. Here we see a coagulation and precipitate formation. This means that Staphylococcus aureus is positive. Let's dispose of our slide biohazard sharps waste container. Now we can move on to our next test. Our next test is the Oxydase Test. The Oxydase Test is used to identify bacteria that produce cytochrome C oxidase. Cytochrome C oxidase is an enzyme of the bacterial electron transport chain. Note, all bacteria that are oxidase positive are aerobic and can use oxygen as a terminal electron acceptor in cellular respiration. This does not mean that they are strict aerobes, however. Bacteria that are oxidase negative may be anaerobic, aerobic, or facultative. The oxidase negative results only means that these organisms do not have the cytochrome C oxidase that oxidizes the test reagent. They may be able to respire using other oxidases in the electron transport chain. Let's begin our experiment. We will first click on the box of sterile sticks and select a stick, which will become our mouse. Then we can choose a colony of Yersinia pseudotuberculosis to place on the dry slide. We will use the stick with the bacteria to smear the bacteria onto one quadrant of the dry slide. We do not see a color change to purple, so this bacteria must be negative for oxidase. Let's dispose of our stick and choose a colony from Pseudomonas aeruginosa. Be sure to choose a colony that is out by itself. We'll use our stick to smear a small amount of the bacteria onto another quadrant of the dry slide. Here we see a color change to purple, indicating that this bacteria is positive for the oxidase test. Let's dispose of our stick in the biohazardous waste. Next, let's choose a colony from Pseudomonas putida. We'll choose a colony that's out by itself and smear a portion of the bacteria onto one quadrant of the dry slide. Again, we see that purple has appeared, meaning that this bacteria is positive for oxidase. We'll dispose of our stick in the biohazardous waste, along with the dry slide. Great! Now we can move on to our next test. Our next test is the Rapid Strep A test. The Quick View Plus Strep A test kit is used to detect Streptococcus pyogenes from a pure culture or directly from a throat swab in a lateral flow immunoassay. In this exercise, we will test two different swab samples. Let's begin. We'll begin by clicking on the clear tube and place it in the test cassette to the tube well. We'll use our reagent A and add it to the tube in our cassette. We will need to squeeze 4 drops of reagent A into the tube. Now we will repeat this using 4 drops of reagent B. Let's test our throat swabs. We will place the swab in the tube and mix for 1 minute and squeeze out the excess liquid against the side of the tube. We'll want to be sure to dispose of the swab in the biohazardous waste container. Now we can place the cap on the tube and place 2 drops of the sample from the tube to the round sample well of the test cassette. Then we'll need to dispose of the tube in the biohazardous waste container. Now we can observe the cassette and watch for the appearance of vertical and horizontal lines in the 3 windows. The first, or far right, is the test window. A positive result will yield a pink vertical line over a blue horizontal line to form a plus sign. Negative would be a blue horizontal line to only form a negative sign. The second window, in the center, is the positive control window, and it will have a pink vertical line only, indicating that the test is working properly and will detect a Strep A antigen if it is present in the sample. If no pink line appears in this window, a negative test result would not be valid. The third window, at the far left, is the test complete window, and it will have a blue horizontal line only. If no blue line appears, the test is either not complete or not enough liquid was used in the test. We see our results for our first bacteria is negative. We'll want to dispose of the cassette in the biohazardous waste container. Now we can repeat the procedure for our next swab. We'll add 4 drops of reagent A, four drops of reagent B, and put in the throat swab and mix for 1 minute. We'll squeeze out a little bit of the excess from the cotton swab into the container. Be sure to dispose of it properly. Next, we can place the lid on the tube and dispense 2 drops of the solution into the testing window and dispose of our biohazardous waste. Now we can incubate for 5 minutes and see what our test results are. We see that this test is positive because we see both a blue line and a pink line overlapping to make a positive sign. Dispose of your test properly. And now we can move on to our next test. Our next test is the Latex Agglutination test. The Latex agglutination technique is used to distinguish different Streptococcus species from one another based on the Lancefield serotype groupings. Let's begin our experiment. We're going to place 1 drop of each of the latex conjugated antibodies to strep antigens A, B, C, F, and G on the ovals labeled 1 through 5 on the sample card. Once all 5 latex reagents are on the card, we can click on the sample that we wish to test. We can test the control ABC, control FG, unknown 1, or unknown 2. Let's do our control ABC. We can choose our sample by simply clicking on it. This will cause a drop of the sample to be dropped onto each oval containing the latex reagents. Next, we'll click on the card that will enlarge it and bring it into view in a 50-second timer. We will hold the card and gently rock it for 15 seconds. Let's look at our results. The first 3 samples show agglutination or clumping. Samples 4 and 5 do not. Based on these results, this is control ABC because A, B, and C showed agglutination. We will add our reagents again to test our next control. This time, let's choose the FG control. We will gently mix for 15 seconds. The first 3 wells are negative. The wells for F and G are positive, just like we would expect for our control FG. Now that this card is full, let's dispose of it in our biohazardous waste and set up for our unknown number 1. Now we're going to add 1 drop of our unknown sample number 1 to each of these wells. Now, let's gently rock our slide for 15 seconds and look at our results. Here we see that well number 3 shows agglutination. This test shows us that we have strep C in sample 1. We have 1 more sample to try. We'll add our 5 reagents to 5 new wells. And add our sample number 2 to each of these wells. Now we'll gently rock the slide for 15 seconds and see our results. Here we see that the first well has agglutination. This means that we have strep group A in our sample 2. We'll be sure to dispose of our cards properly. Now we can go on to our final test. The final test is the GonoCheck 2 test. The GonoCheck 2 test is used diagnostically to identify pathogenic Neisseria species in gonorrhea from N. meningitidis in samples, distinguishing them from commensal Neisseria and closely related Moraxella catarrhalis. Let's begin our procedure. We will first begin by sterilizing our inoculation loop by passing it over the Bunsen burner. We will place the loop in the flame until it is visibly red hot and then allow a few seconds to cool. Now we can drag the loop to one of the 4 petri dishes and pick a colony. We will add the bacteria from the loop to one of the 4 test tubes on the bench. We must decontaminate the loop before placing it on the bench top. We must do this by flaming the loop once again. Now our tube reappears with a choice of 2 caps to place on the tube. We are going to click on the clear cap, which will move to the top of the tube, and the timer will begin to count down. When the timer stops, the sample is yellow due to the hydrolysis of glutamyl p-nitroanilide by glutamyl aminopeptidase. N. meningitidis is the only one of these 4 that produces this enzyme. Let's pick a colony from our next plate and repeat the procedure. We'll want to make sure to decontaminate our loop before placing it on the bench. We'll incubate our sample for 30 minutes. Here we see a color change to blue, indicating that this is N. lactomica. Blue coloration is due to the hydrolysis of 5-bromo-4-chloro-3-indole D-galactopyranoside by galactosidase, an enzyme involved in lactose fermentation. N. lactomica is the only Neisseria that ferments lactose. Now we'll choose a colony from our next plate. And place it into our next tube. And decontaminate our loop. So we can place it back on our bench. We'll now incubate this sample for 30 minutes. We see no color change, so we will select E to continue with the next test. The clear cap will come off the tube, and you must first discard it in the biohazardous waste container. The red cap is then placed onto the tube by clicking onto it. Now, we can invert and mix the tube. We still observe no color change. This indicates that this bacteria is Moraxella catarrhalis. Let's flame our loop to get our last sample. Now we'll choose a colony from our next plate. We'll choose our colony and place it into the last vial. We'll flame the loop once again so we can place this safely on our bench. Now we'll place the white cap and incubate for 30 minutes. We are unable to identify the test, so additional testing will be needed. Let's discard the white cap in the biohazardous waste and place the red cap onto the tube. We will invert it and mix. We observe a color change to red, indicating that this bacteria is in gonorrhea. Congratulations on finishing your last task. Now you can complete the laboratory questions. Thank you for watching.